parent, brought together to produce a new set of instructions and reshuffled in each generation. But sometimes these instructions carry errors, and these can create problems. Faulty genes can cause serious illnesses. Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we've made a great leap forward. This increasingly accurate new technology is faster to use, relatively simple, and cheap compared with previous methods.
RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers have deactivated one or both of Cas9's cutting domains using new enzymes.
Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA.
It's an okay way to maybe knock out a gene, but it's not the way that we really want to do genome editing. The second repair pathway is a lot more interesting. In this repair pathway, it takes a homologous piece of DNA, and now mind you, in a diploid organism like people, we've got one copy of our genome from our mom and one from our dad. So if one gets damaged, it can use the other chromosome to repair it. So that's where this comes from. The repair is made, and now the genome is safe again. The way that we can hijack this is we can feed it a false piece of DNA, a piece that has homology on both ends, but it's different in the middle. So now you can put whatever you want in the center, and the cell gets fooled. So you can change a letter, you can take letters out, but